For a long time, man has taken the earth for granted, believing that we are not capable of destroying our own world. The facts, unfortunately, do not agree. A direct correlation between CO2 emissions, rising global temperature, and a rapidly increasing number of polluted scenes like this one mean that we can no longer afford to be complacent. Time really is running out. The world is in an energy crisis and something has to be done. With a problem so huge, you can be forgiven for believing that the solution itself must also be huge. In fact, the potential answer to Earth's biggest problem today can be found within a bacterium that's around 2 micrometers long. A group of enzymes found within E. coli have certain physiological properties which may allow man to one day harness efficient production of renewable fuel and bring global warming to a halt.
Well, let's start from the beginning. E. coli is a gram-negative, rod-shaped bacterium, most commonly found in the lower intestines of warm-blooded animals. Although most strains are harmless, there are a few which are capable of making us ill. E. coli is probably the most widely studied prokaryotic organism in the world. We can put this down to the fact that it can be easily cultivated in lab conditions and is relatively inexpensive to work with. You would think that with all the recent research that has gone into this tiny microorganism that there wouldn't be much more we could learn from it. However, there are still many secrets that it holds. One of the most interesting recent developments is the study of particular enzymes called hydrogenases. They are found within the bacterium and are capable of essentially converting hydrogen from the atmosphere into an energy source.
This is Frank Sargent. He's a professor of bacterial physiology here at the University of Dundee who has done a lot of work with hydrogenases. During this video, we will be talking to him about his contribution to a published paper about these enzymes, as well as the broader possibilities in the world of biofuel that this research has opened up. But first, let's break down some of the core science from the paper. In E. coli bacterium, there are 3 types of enzymes known as hydrogenases. However, only 2 of these are actually involved in the uptake of hydrogen. These are appropriately named hydrogenase 1 and hydrogenase 2. In broad terms, the general function of a hydrogenase is to either produce hydrogen or break it down into its constituent parts. There are 3 distinct types of hydrogenases known to science, each defined by the metal ion found in their structural active site. The most common types consist of a combination of iron and nickel ions, and this is the type of classification which both hydrogenases being studied fall into. The research for this paper seeks to determine how the functions of these 2 specific enzymes contrast, along with the different environmental conditions which they are capable of working under. Many advanced lab techniques are used to acquire these findings. The main one is protein film electrochemistry. This process involves a thin layer of enzyme being adsorbed onto the surface of an electrode, which can then be completely controlled in terms of precise concentrations of gases that the enzyme is exposed to. This allows the environment to be changed to varying degrees of aerobic or anaerobic conditions. What findings did this paper unveil? Well, who better to talk to than the man himself, Professor Frank Sargent.
So how did you initially get involved in this investigation? You know, what was kind of the reason why you chose these hydrogenases?
Well, this goes way back actually to when I was a student, and I'm thinking about when I finished my 4th year and I wanted to do a PhD. I got really interested in enzymes that contain metals. So I looked around the UK for labs that worked on metalloenzymes, and there's actually one in Dundee that worked on nickel-dependent hydrogenases, which actually I didn't know anything about at the time. And then I came to learn more about it, and actually I've always, from those days, that was 1992, I've always studied hydrogenases since then.
Since 1992?
Yeah, so it's one of my favourite enzymes, probably because it's got lots of different applications. You can study it if you're into proteins, you can study it if you're into metals or into physics. As you've seen in this paper, there's lots of complicated physical techniques, or you can study it if you're into biofuels and bioenergy. So it's, it meets a lot of interest in the sort of things that I like to do.
So you know a thing or two about them? Well, the paper obviously focuses on hydrogenase.
I know it.
Obviously it focuses on hydrogenase 1 and 2. Could you briefly give us the difference between the 2 of them?
Yeah, so it's interesting that so you'll often read in textbooks that bacteria just have one gene, they have one chromosome and they have one gene. And then a human cell has got 2 chromosomes and 2 genes for everything and stuff like that. But actually bacteria are more complicated than that, because they tend to have more than one gene for similar enzymes scattered across a single chromosome. And what E. coli has, it's got a gene for hydrogenase 1, which it expresses in late stationary phase, and which it co-expresses with a cytochrome oxidase. And I think it's there and it's an oxygen tolerant enzyme, so it can operate in air. And I think it's there just in case the environment changes and E. coli moves from anaerobic to an aerobic environment, and then it can still use hydrogen under those conditions. Hydrogenase 2 is expressed in a completely different condition. It's not expressed in the stationary phase at all. It's expressed in the early stages of anaerobic growth, and it's used to generate a protein motor force, which gives it energy to make ATP. So they're expressed at different times. One of them can withstand oxygen attack, the other one can't. So they have slightly different properties. So E. coli is clever in that way, and it modulates the expression. What's the potential? What's the point of this paper?
What's the point of this paper?
Well, this is an important paper you've picked because this was really the first paper that showed that hydrogenase 1 could operate in the air. And that was a very specialist sort of discovery that a few scientists were interested in, but it has huge impacts for society at large. Because what we've been able to do since, we've been able to take this little protein, and what it does, it produces it. You can attach it to an electrode, and you can put electricity in, and that will make hydrogen. So we've been able to make almost like a battery out of this protein mixed with electrodes, that will produce hydrogen, or can produce electricity in the presence of hydrogen. So it's the start of a whole new way of making clean, cheap, non-toxic fuel for the future. So it has implications for society at large, and this is why the government have decided to fund this at a high level, because they can see that the underlying science is really important for finding new energy sources in the future.
So, let's go through some of the most interesting results of the paper. This graph essentially shows that hydrogenase 1 is oxygen tolerant. Now the enzyme is adsorbed onto the electrode and has electrons fired at it. Now because the atmosphere contains hydrogen, as the potential begins to increase, as does the enzyme's activity, which is measured in current. Now when oxygen is injected, you can see the enzyme activity plummets right down to near zero. However, it manages to recover and completely recycle back to 100%. When this is compared to the graph for hydrogenase 2, we can see that enzyme activity goes to a fairly similar level until it is hit with oxygen. Now, when this happens, the activity plummets down to zero and shows no sign of recovery. The reason hydrogenase 1 is oxygen tolerant is because when oxygen hits the active site, it binds with hydrogen and forms water. This essentially means the enzyme is working backwards. However, it gives it time to recover and allows it to again bind with hydrogen. In the case of hydrogenase 2, however, when oxygen hits the active site, it forms a complex with nickel, meaning the enzyme can no longer bind to hydrogen and rendering it useless. So, in summary, with oil levels sufficient to last us less than 100 years, it is imperative that we develop alternative sources of energy. The study of these two enzymes are at the forefront of research in the hope of making hydrogen fuel production more efficient. Now, the University of Dundee is one such institution attempting to do so, by offering PhD opportunities studying hydrogenase in relation to the production of biofuels. Everyone needs to realise the stark truth that our use of carbon fuels is not only finite, but is also destroying the planet. We will one day hopefully master the production of greener fuels and put an end to this planetary turmoil.